Thanks everyone for coming today for the FBO, my first graduate student, Dan. And he worked on new nanoscale materials, but then transitioned to more biological approaches. And through friends and collaborators, he was able to bring biology into the group, and then worked at the interface between the two, which is, I think, a pretty interesting area in both sensing small mechanical forces in biological systems, but also actuating them as well. And so I'll let the work speak for itself, but I'm excited today to welcome Dan to present the stuff he's worked on for the past 5 years. So thanks a lot.
Thank you all for being here today. It's my pleasure to present the research that I've been doing in the last 5 years. My talk describes an effort to develop a very new concept, a very new field of biointerface nanopiezoelectrics for the important understanding and application of biological deformations. So let me start my talk by telling you how important deformations are in biological systems. From a microscopic and bulky organs to small individual cells like heart cells or neurons, deformations are everywhere. They are inherent to cellular functions and physiology. So life stops when the heart stops beating or brain stops functioning within no mechanical deformations in the nerve systems. So deformations are signals of life. But they are also signal of diseases. Scientists in cancer research, for example, found that cancer cell has a much higher deformability than the normal cells. So sensing this deformational difference could offer a valuable approach for cancer. But besides of the sensing applications, we can also use deformation to engineer cells. One of the typical examples in a paper in PNAS this year showing that flowing the cells into the microfluidic channels and squeeze them, the scientists can deliver easily drugs into the cells and engineer them. But now let's go to a more specific but very important system, is the neuronal systems. It's very well known that neurons communicate to each other by sending and receiving electrical signals named action potential. But very less is known about the deformational signal associated with this action potential. Well, in fact, scientists a long time ago found that the Krapf nerve tissues also deform and expanding and apply addition force at the same time with action potentials. But this coupling between electrics and mechanics is not to appear in neuronal systems, but actually the intrinsic property of a cellular membrane. So using an artificial membrane made up from the lipid molecules, the group in Russia in the years of 90s showed that the membrane can flap under the applied voltage, and they also can polarize under the deformations. So the explanation for that is based on the model of liquid crystal. So the cellular membrane has the structure like the liquid crystal. Each of the lipid molecules has the intrinsic dipole. So in the flat and non-deformed membrane, imagine, you have non-net polarization because the random distribution of the dipole in the structure. But when you bend it, because this molecule has an asymmetric shape, now they reoriented, and there's more net dipole direct into one direction. And that's the mechanism that the membrane can polarize under the deformations. So it's very easy to see that the smaller cells with a higher curvature could exhibit a more significant coupling between electrics and mechanics. But so far, the only tool which are able to probe these deformations in a very tiny cell is just the IFM, which is stand for atomic force microscopy. So in a paper in Nature, Zhang and the other co-authors showing that using the micropipette and AFM, they can probe the coupling between the electrical and mechanical signals at the same time. And this is not the neurons, this is the kidney cells. It's just showing that the coupling between mechanics and electrics is universal. So in each of the electrical stimulation into the cell, they can see that the cell deformed by looking at the deflection of the FM tip. But the problem for the FM is it using a very small nanotip which can punch a hole into the cells and can be very invasive. It also produces non-electrical outputs, which is very inconvenient and it limited the method from scaling up to a large multiplexing measurement on multiple cells. So that's why there's a need for finding an alternative approach, an alternative materials which can interface effectively with cells and capture the deformations in the cellular structures. So the material should have two requirements. First, they should transform the deformations in cells to the electrical outputs, which can be analyzed and can be sensed by human, by us, and by computers. But the second requirement is the material should be small because it has to make a size commensurate and the conformal interfaces with the small and curvy cells. So this second requirement means that the material should be smaller and ideally should be built in the structure like the nanomaterials. And that's why the concept of bio-nano interface comes from. The scientists realize this benefit of the nanomaterials, and that's why they develop a different nano tools, for example, like silicon nanowires or graphene to interface with cell and send very small cellular signals. The nano tool also has a benefit because it has a very high sensitivity due to the high surface ratio to the bulk volume. But none of these nano tools was able to detect the deformation signal in cells, and that's why there's still a need for finding a biomechanical nano probe. So now the solution for that, as I recall you, the first requirement is the material should be able to perform the transduction between the mechanical and the electrical energy signals. And that's exactly the definition of the piezoelectric materials. And also second requirement say that we should have this material as a nanoscale, which is the piezoelectric nanomaterials. And so in the next slides of my talk, I will describe to you our effort in the generation of these piezoelectric nanomaterials for the application as a mechanical nanoprobes to sense cellular deformations. So let me first describe the fundamental of the piezoelectric effect. So let's get an example. The most common materials, the piezoelectric material is quartz, which is basically silicon dioxide. It has silicon at the center and surrounded by the oxygen atom. So in the normal condition, there's no dipole inside a unit cell of quartz. But when you put it in the compression or tensions, because this oxygen atom doesn't have the non-inverse symmetric atom around the silicon, now a dipole is built up inside the unit cells of quartz, and over across single crystalline silicon dioxide, you have a net polarizations. And that's the mechanism why the piezoelectric material can polarize under the deformations. So looking at the theory of the piezoelectricity, the most important parameter that keep in mind it is the D33. This is the piezoelectric constant. The number 3 just stands for the Z direction, that we simplify the system by looking at one direction of the polarization and deformations. So D33 appear in the coupling equations. We tell you how much polarization the material has under the applied stress T and how much strain that the material can deform under the applied electrical field E. And from that equation, you can calculate the electrical energy stored in the piezo element, which is E is equal to 1/2 of capacitance times square of the voltage. And you can derive the E is proportional to the applied stress, the volume, and most importantly, is proportional to D33. And now you can also calculate the input mechanical energy, which is 1/2 of stress times strain times volume. And then you get E divided by that input mechanical energy, you get the conversion efficiency K is also proportional to D33. So D33 tells you everything. And if you're looking at the common piezoelectric material, in the bulk piezoelectric material, it wouldn't be difficult to see that PZT, which stands for lead zirconium, that has the outstanding D33. So this nanomaterial of PZT, that's why it's really attractive, and people try to make the nanomaterials of PZT. But one of the possible explanations for this enhancement of D33 is based on the crystallized structure. So PZT has a perovskite structure. So similar to PZT, a less complicated composite material like PZ and PT also have the same crystallized structure. So if you, for example, if you're looking at the PZ, which stands for laser chloride, because the rhombohedral crystal structure of the PZ, now under the deformation, it has a totally 8 possible orientation to polarize towards 8 corners of this crystal. And that's why the coin has a possible 8 possible domain state under the deformations. Now similar to PZ, PT has totally 6 possible domain states. When you mix them together in 50-50 percentage, you make the material confused. And now, interestingly, the composite material PZT lies at this morphotropic phase boundary, has a totally 14 possible domain states. And that's explains why the material has an enhancement in the D33. So to get a good PZT nanomaterial, we should get 3 conditions. First, it should have a good stoichiometry, a good composition. Second, it should have a crystallized structures. And third, it should have a horizontal alignment, just for the purpose of electronic integrations. So besides of the top-down process, which usually relies on very complicated and expensive beam lithography, looking at the current methods in the bottom-up approach, all of them has limitation. For example, VOS is very difficult to create some complex composition material like PZT, because it requires a special catalyst which can form a supersaturate liquid phase with PZT and has a phase separation. In the second method, Sonegel has the constraint that it only creates polycrystalline PZT nanomaterial thermal process. It uses very high temperature to create the single crystalline PZT nanomaterial in solution. But it has a slow kinetics and also requires expensive autoclaves. Furthermore, you can see that it creates a random distribution of the PZT nanomaterials inside the solution. Now, we need a further step to collect them, align them on the horizontal surface for the electronic integrations. So that's why we came up with our own idea of applications. We asked ourselves, can we simultaneously control the composition, the crystallized structure, and the horizontal alignment of PZT nanomaterials? And the idea, based on the achievement that people already had, they already make a high-quality single crystalline PZT film. So why don't we use that to scale it down to make these nanomaterials out from this high-quality single crystalline film? And this is a schematic that describes what we did. So we have a very high-quality PZT film epitaxially deposited on MgL substrate. And now, if somehow we can have a metal nanowire, something very hard nanowire like nickel nanowire on top of the PZT film, then we can use the dry etching to transfer that nanowires into these PZT nanowires. And the way we do that is the method named pencil. So pencil just relies on a very low resolution photolithography and a very simple electrochemical etching process. So this is a schematic describing that process. So we have a nickel film. We can pattern the micro-resist slide on top of it by using photolithography. And we can use electrochemical etching to scale down this film into a small nanowires, because we have a very good control in voltage and time of the etching. And then we remove that photoresist slide, we can have the nickel nanowires. So the method is simple, based on the standard technology for making IC circuit. And we avoid any damage on the substrate by the gentle electrochemical etching process. And we don't use any complicated or expensive in lithography. And this is the result showing you that we was able to make a large array of PZT nanomaterials on a 3 and 4-inch wafer scale. We can scale it down to 40 nanometer nanowires, or with a different shape, like a ring or curve shape. They are very important for the optical applications. But the method is also powerful because it not only makes PZT nanowires, it can also make, and actually we did make chrome, nickel, silicon or silicon dioxide nanowires. We can go further and smooth these multi-nanowires by just one single laser pulse by a method named SPEL that we adapted from, developed by Professor Stephen Chow in electrical engineering. We can shine a laser pulse on these nanowires and melt them in a very short time of the laser pulse. And when it's melted, it flows like a liquid, and it can minimize the surface energy. And now it can smooth itself through the process named cell perfection. And so you can see that the image showing the nanowires after the laser and before the laser show a significant enhancement in the smoothness of the nanowires. But most importantly, we measure the piezoelectric constant in the PZT nanomaterial. And the way we did that, if we use a PFM, which stands for piezo force microscopy, this is a modified AFM in which the tip is conducting. Now we can apply AC voltage between the tip and the bottom and getting the deformation with the same frequency via the locking amplifier. Now we can see that when we increase in the AC amplitude, we can have the increasement of the deformations. And the D33 we get is about 140 picometer per volt. At the time I published this paper, this is the highest D33 of piezoelectric nanomaterials. So it's a really wonderful result. And PZT indeed present a promising platform for being a nanoprobe to probe the mechanical deformation in cells. So in the next slide of my talk, I will describe to you our work in interfacing this nanomaterial with cells and measuring the deformation in cells. But the first question we ask before we do anything is if this material is biocompatible, because PZT is well known with lead, which is very toxic. And so we use a cell, a PC12 cell line, which is a stem cell that if you feed it with the nerve growth factor, it can differentiate into the neurons. Now we culture it directly onto the PZT nanoribbons. And the first time we saw it grow as normally as the neurons, but we also do the electrophysiology test, and we're injecting the current into the cell, and we see that the cell increasing the transmembrane potential, and at some threshold, it can trigger the small spike, look like axial potential. So looking at the literature and compare this result, we see that the cell indeed behave normally in the electrophysiology test. But we also did a thorough experiment, and it surprised us that for a long time, even weeks, the cell, compared to the cell culture on the standard culture environment, it has really not much difference between the cells culture on the PZT nanoribbons and the cell culture on the standard environment. So it's showing that yes, the PZT indeed has a very good biocompatibility with cells, at least in the time scale, on the time scale of weeks. So that's help us to keep going on and develop this nanoribbon chip, which can sense the deformations in cells. So this is schematically describe the nanoribbon chip that we developed. We somehow create the suspended PZT nanoribbons, and we can put the pipette on top of the cells, and we can inject the electrical currents into the cells and see the deformation out from the cells. And the way we did that is we have a PZT nanoribbons on top of the MgO substrate, like the conventional method that I just presented. And then we can parent the ITO electrodes. We can passivate the electrode by using the insulating layer of silicon nitride and suspending the PZT nanoribbons by using phosphoric acid to attenuate the MgO. So that's the results of our developed chip that you can see arrays of the suspended PZT nanoribbons. Now we can put the cells on top of it. And this is a wonderful image that we're really happy to have it. And then we can go further and do the sensing measurement, which is you can see that the chip is very, very transparent, that we can see the cells at the same time with the micropipette. And now we can manipulate the micropipette, put on top of the cell, inject the current into the cell to stimulate the electrical signal on the cells, and in each of the electrical signal, we can see a beautiful coherent piezoelectric signals come out from that. So it's a wonderful result, but we also want to ask what is the fundamental going on here. And so the work we collaborate with the professor in UPenn, Professor Prashant in UPenn, that we based on the model that Zhang just already reported in the paper in Nature in 2001, that the theoretical model is very long analysis in the force that the cell apply onto the piezoelectric ribbons. But without going deeply into the details, I will tell you the two important equations here that describe what's going on in the system. The first equation is the Lippmann equations that it tell you because of the charge distribution around inside and outside of the membrane. Now that detects the membrane tension on the cells is similar to actual wetting phenomena, that if you have a liquid dropping on top of the subject, and you apply a voltage into the droplet, it will spread in the droplet because you change the membrane tensions. So Lippmann is the first one who developed these equations, and we can apply that for the bilayer lipid membrane to derive the relation between the voltage applied on the membrane and the membrane tension taus on each side of the cells. And
could I just ask a question?
Yeah.
Your experiments are done in an atmosphere of
No,
damp air, or
right,
not in a fluid?
In the fluid, yeah. The cell not gonna alive if we do it outside of the growth medium.
Yeah.
So yes, everything is in the liquid. Yeah. So and without the liquid, you don't have the charge around in the cells.
Yeah.
So it's an ionic solution. And the second equation, which is also important, is the Laplace equation, which is telling you that the membrane tension tau detects the radius of the cell via the pressure difference between the inside and outside of the cells. So because the cell is healthy, now we keep this pressure constant, you would see that when you change the membrane potential, you would change the membrane, you could change the membrane tension tau, and then you're going to deform the cell by changing the radius. And doing the analysis on the force and make some approximation, we can get the force, which is F, is going to be related to the radius, to the membrane tension tau, and finally it would depends on the voltage on the membrane V. So from this equation system, you can derive the theoretical force upon the applied voltage onto the cells.
So what is that? What force is it?
This is the force that the cell apply onto the piezoelectric, suspended piezoelectric ribbons. By deforming, and assuming that the cell is deformed very small and the PZT ribbon is very stiff compared to the cell.
Are you assuming anything about the contact interaction between the cell and the PZT? I mean, if there's very little frictional force, for example, I mean, it can, in principle, it can deform any way that it wants, and it's just going to be displaced as a
right. So in this
body.
experiment, right? So that's a good question. So in this experiment, we have the pipette tip on top of the cell. Now we can confine upward deformations, and most of the force now applied underneath of the cell and on the very thin, super thin piezoelectric ribbons. Yeah.
But why isn't it, when you apply the current,
Yeah.
and you've got a very hard boundary up top and a pretty stiff boundary on the bottom,
Do the calibration on the cellular force that the cell apply onto the PZT ribbons upon different membrane voltage depolarization. And compared to the theoretical data from the last slide I showed you, it shows a really good fit between the theories and the experiments. And we can go further to calculate, to estimate how much deflection the cell indeed have on the PZT nanoribbons. And using just a simple beam theory, we can get a very small deformation, about 1 nanometer from this one single cell upon 100 millimole membrane depolarizations. They show you the very small deformations, and the PZT indeed has a very high sensitivity to sense the deformations.
So if we go further, so that's a fit. So what you call theoretical data is
Yeah.
some fitting parameter based on the expression on the last slide.
Right.
So what does that tell you about the cell? Right? I mean, suppose that I measure that data, so I have a fitting parameter. So what does that fitting parameter tell me?
the fitting parameters. So it's a good fit. So it's telling us that the model that it explains what's going on in the experimental data, and somehow it explains why when you increase the magnetic voltage that you can increase the cellular force or the deformations.
I think you're making a strong statement. So your statement is that because it fits the data, therefore it's a good model for describing it. I think you're making an overstatement. It does fit the data, but I can come up with a million different models that fit the data, but they don't have any physical basis.
Right.
So that's why I was asking you about the fitting constant, that if you have a reasonable understanding of what's going on, you should be able to estimate
Right.
what that constant is. So how does it compare to the fit?
So in this model, actually, one of the prediction of this model is if you're changing the ionic solution, now you can change the direction of the deformations. So if you put it in like the non-ionic solutions, that actually confirmed by the experiment that I showed you in the Zhang papers. This model actually is not really a new model because we based on the work of Zhang in Nature's, and they actually do a lot of prediction on that, and we only do the analysis on the cellular force and compare that to our piezoelectric Riemann force. But yeah, well, the significance of this model is they can predict a lot of things, and they do changing the solution, changing the putting in like drugs and something to see if this model it does predict the phenomena of this deformation. So it's not a really new model in our case because we based on 95% of this model is in nature. So yeah. Okay, so but we also want to show that the PCT in our ribbons is not just able to sense a single cell. It can also be scaled up onto a large area by using a conventional method to transfer the PCT arrays onto a polymeric PDMS, flexible and stretchable polymeric substrate. We can interface this PCT in our ribbon chip with the electrodes, with the GO electrodes on the columns, on the tissue of the columns. So when the column deforms in a mimic respiratory process, it deforms the chip, and it can generate the output electrical signals. And this is the result that you can see that in each of the deformation of the column, there's output electrical signal come out from that, and the voltage is about minus 0.6 to 0.6 from a very small strain from these deformations. They're just showing that the PCT now ribbon is very powerful because it's not just able to sense the mechanical signal from one single cell, but it can also sense the deformation from a microscopic tissue of the multiple cells. And this application also very useful if you think about using that to get the output power and to power the electronic devices. So I've been talking about one side of the piezoelectric applications, which is using the transformation from mechanics to electrics for sensing application. But the PCT also has the capability to be an actuator. That means we can use electrics to control the material, make the material deform, and apply the deformation into the cells or tissues to engineer their behavior via mechanical effects. So in the last part of my talk, I will describe to you an achievement that we make as a proof of concept that the mechanical effect indeed can induce the growth of neuronal cells in a very small microchannels. So it's no need to talk again about how important the brain is in human bodies. So brain makes up of billions of neuronal cells, and in each of neuronal cells has three main parts. The dendrites part is the one who receives the signal. The cell body is to process the signal, and the axon is the most important part which transports the signal. So the neurons in a brain connecting to each other in a very directional and complex circuit to control the brain activity. They have polarity because the signal only flows in one direction from dendrites to axon.
Say that again?
So it has a polarity because the signal flow only in one direction from
What do you
dendrite
mean by
to the axons.
the signal flows only in one direction? That's what I don't understand.
Right. So
So you can tweak one end and the information arrives at the other?
exactly, yes.
That's all, but how does that information transfer?
So that transfer by sending and receiving the electrical signal, the action potential,
Why is that? Where's the rectifier?
the rectifier.
Well, if it only sends it in one direction, there has to be some rectification, no? Why isn't it symmetric?
Well, actually, some of the neurons has a dipolar direction. That means it can send in two directions. Most of them in one direction. Let me just clarify that. And because of the difference in the microscopic structure of the neurons, so the dendrites usually have the acting. So that actually is a really good question too, because people don't completely understand why that happened. And they're actually looking at the single neurons and stimulate them and see why the signal flow from one side to the other side. But the common explanation is based on the microstructure of these cells. The axon has a lot of microtubules, which usually is the main thing to transport the signal. And the dendrites, so you see the structure of the neurons by itself has a lot of dendrites which can receive the signal. The axon is the one who send out the signals.
Well, don't get hung up on this point.
Okay, yeah. I can go back because that not
We can
completely
look at it later.
Yeah, display. Okay. So entire neuroscience field was actually try to understand one thing: how this circuit of the neurons form, how they organize, and the signal, why they flow in one direction, like you said. So the way they usually do that is they take a part of the brain out from the human body. They stimulate the cells and see how the signal flow in that brain slice. But doing that costs a lot, as you can imagine. And so the alternative approach is they try to create this neuronal circuit in vitro in the lab. And so they can stimulate them and see and understanding how this circuit is organized, and then they can imply to the in vivo image, and then they can do further research inside of the human body. So the way to do that is they need to find a method to control the growth direction of the neurons, to create a directional neuronal circuit in vitro. But if we can find a way to control the growth direction of the neurons, it also brings about a lot of applications. One of these is the application in regenerations of the damaged axon on neurons. Imagine if you have a car accident. It's really bad because it can break the neurons, and now you can be put in the memory loss of the paralysis. And now the way the scientists solve this problem is usually they use the neuronal stem cells, or they try to find some method to initiate the growth of, regenerate the growth of these axons, or control the growth direction of the stem cell into this position. So that's the need of a method which can initiate the growth and the control of the growth direction in neurons. So again, we need to look at the current methods that people already have to somehow control the growth direction of the neurons. They either can pattern an adhesive layer on the microchip, or a microstructure on the substrate which can enhance the adhesion between the cells and the substrate. So here's a CNT carbon nanotube that's showing that the cell adhere into this part more than the original tissue substrate. Or as a biomolecular perspective, people also use the drugs. We can guide the axonal growth by using some special drug. But all of this has a limitation, and it's low yield because it can adhere a very small number of cells onto this adhesive layer of the microstructure.